So before I start, I actually want to thank the people that actually do the work. I get the privilege to talk about it and hang out with the wonderful people here. So thanks so much, and also to these funding agencies that have been supportive of us. So as a graduate student, in my undergraduate, I'm a chemical engineer by training. I came across this paper by Harrington and coworkers. And what we can see are three patients. The first is a patient with a tumor at the base of the tongue, a patient with breast cancer and metastases in the local lymph nodes, and a patient with lung cancer and metastases in the bone marrow. And actually, each patient then has a different type of cancer, but they were all treated with a similar treatment, 100 nanometer particles that were injected intravenously into the vein. The nanoparticles circulated in the body and then accumulated at the tumor site. And that's why you can actually use different imaging modalities to image the tumor itself. So this is targeting when we talk about it in cancer. And it's very advantageous because if you have a chemotherapy or another type of medicine inside the nanoparticle, instead of treating the entire body, it actually focuses at the disease spot. So this is real. This is clinical today. And the reason it actually works is interesting in cancer and infections and inflammation is the blood vessels we have at those sites actually differ very much from the blood vessels we have in healthy tissues inside the body. So in a healthy tissue, if this would be a nanoparticle in white, all these dots here, the blood vessels are a little bit like a pipe that actually has no holes in it. It's a continuous pipe. And the nanoparticles actually cannot exit the blood circulation and enter the healthy tissue. Contrarily, in the sites of cancer, sites of different types of disease, but primarily cancer, the blood vessels are actually discontinuous. They're like pipes with small holes in them, actually nano holes inside of the sides of the blood vessels. And a nanoparticle traveling in the circulation can exit the circulation and enter and penetrate and get trapped in the disease site. And this actually gave birth for an entire field that different types of technologies were capable of actually targeting specific sites inside the body and accumulating a medicine in one area, in a diseased area, rather than affecting healthy areas in the body. In fact, to date, nearly 250 nanotechnologies have been approved by the FDA. None of them actually has nanotechnology listed on the label. For example, some of them we know it because we can actually buy the medicine, test it under the microscope, and see that there's nanotechnology inside. But in most cases, the FDA actually keeps a secret together with the company. And it's a little bit like Coca-Cola, that we know the ingredients, but we just don't know how the ratios between them and how they are arranged. In nanotechnologies, many times, because the nanotechnology adds another functionality such as targeting, as we saw earlier on, the FDA will not disclose this to the users themselves, and but it does record it within its trials. The first nanotechnology to reach the market is called Doxil. It's a 100 nanometer particle with the anti-cancer agent doxorubicin inside of it, and the advantage that actually granted is, one is targeting, and two was it reduced terrible side effects this chemotherapy had on the heart of patients. It was invented in Israel and approved in 1992. And since then, many others have actually changed the space of cancer therapy. So when we talk about a nanoparticle, usually we'll be imagining, or at least I imagine, in my mind it's a tennis ball that has a medicine inside of it. And in the case that we work in many times is a lipid-based tennis ball. It's called the liposome. And there's a lot of engineering in each one of these systems. For example, if you load a hydrophilic agent into the aqueous core inside this nanoparticle, you want it to leak out only when it reaches the disease site. So you'll have to tune the membrane of this particle so that it actually is impermeable when it's in circulation, but it becomes permeable when it reaches the disease site, the tumor, and then it has to release the medicine at a sufficient rate so that it's actually active at the disease site. The size of the particle also plays a major role when we talk about treating different types of diseases. For example, when we talk about treating cancer, usually the 100 nanometer range is the right range for targeting most types of tumors. But a MAR who would be interested in targeting the brain, it's usually much, much smaller, say around 15 nanometers, in order to get effective targeting of the brain. The outer surface of a nanoparticle also has to be modified in order to reach or not reach different regions inside the body. For example, if we want to disguise the nanoparticle from the immune system, we'll coat it with a polymer layer that actually absorbs a lot of water molecules onto it, such as polyethylene glycol. The body will see water instead of seeing the nanoparticle, and that will extend the half-life of a nanoparticle in circulation from, say, 30 minutes to 72 hours. Contrarily, if you want to actually have the nanoparticle taken up very rapidly by the immune system, you'll actually add antibodies to the surface of these particles so they are recognized either by the immune system or that they recognize a specific target cell inside the body. And I think a take-home message in nanotechnology is that for each type of disease or for each condition, you usually want to re-engineer your system, maybe not from scratch, but at least think of your target when you come to deliver a technology locally. And just to put things into scale, a nanoparticle is about 1,000 times smaller than any cell inside our body. And in a teaspoon, actually, of nanoparticles, you have enough nanoparticles to target each cell inside an adult 80-kilogram human body. So if we imagine it a little bit like a vitamin pill to our entire body, a nanoparticle should be actually conditioned to treat a specific cell inside the body or many different cells that need to be treated. And again, to put it in perspective, a nanoparticle, we take a hair off our head, and I think many of us are aware of it, and we divide it by about 1,000, we'll reach the size of some of these nanoparticles that are used today in medicine. So going back to trying and condition different materials for different applications, this is an example that the bilayer or the type of particle that's used has to be actually tuned to treat different diseases. Here we're treating a disease of the joint. It's called osteoarthritis. It's degradation of the joints. And the nanoparticles used here actually are soft nanoparticles, so they can reduce friction and wear in osteoarthritic joints. They're constructed in a manner that can actually absorb the body's weight, a little bit like a mattress, and also coat the layers, the opposing layers of the cartilage, in order to reduce friction and wear, and at the end of the day, pain in patients. This is, by the way, is already a clinical tool that's used today. So going back to cancer, in personalized medicine, which is one of the new dogmas we have today when treating patients. What I have in my mind when I think of personalized medicine is house, right? The patient is lying there, and the physicians either know the wrong answer or don't know, and that's sometimes even worse. And I'm not sure. But then house at least comes and saves the day, right? He comes, he sits and meets the patient and knows what to prescribe. However, in reality, we know it's actually very, very different. And if we talk about cancer therapy, specifically if we take breast cancer therapy, at least 30% of the patients are still prescribed an ineffective medication. Breast cancer is probably one of the most studied cancers today, and their technologies are far advanced. And still we have a 30% miss in patients. This is devastating when you're given an ineffective medication. It's devastating to the patient, it's devastating to their families, and it's also devastating to the caregivers that really want to cure their patients. And when we talk about personalized medicine, one approach that is used today is a biopsy will be taken from the tumor or from the patient itself. It will be sequenced for different genetic markers, and then based on that, a drug will be selected for the patient. But I'd want to give you a real clinical example of this type of drug selection. So this was done. This is a real patient. Unfortunately, she passed away some time ago, and she was sequenced by one of the companies, and they gave her choices of different medications. Now you have 6 medicines here. You cannot prescribe all 6 simultaneously. So which one would you choose as a physician? And this is actually the state of the art. These are the best systems we have out there. So even sometimes based on genetic sequencing, we still don't have a full picture of which medicine will work and which will not work inside the patient's body. So that's when a second set of technologies actually kicked in. Biopsy would be taken from the patient. It will be grown in a plate, in a dish. And now in this dish, you can actually screen many different medicines to try and understand which one will be most effective in treating the patient's own cells, so patient-derived cells. But once the cells actually see plastic, they respond differently than they respond inside the body, and it wasn't giving good enough results. So another step was needed to be taken. A biopsy is taken from the patient. It's grown now inside a mouse, so now you have an entire body environment around it, and you can screen different types of medicines. And if this takes 4 to 6 weeks, this takes approximately 4 to 6 months. It's just too long for most patients. And we're not just small or large mice ourselves, so it's not perfect. And we thought maybe nanotechnology could offer some competition in this space. Specifically, what we asked is, could we use nanotechnology to screen medicines inside a patient's body before we start treating a patient? And we got our motivation actually from allergy tests. In an allergy test, you usually don't sequence the patient for different genetic mutations. You just test the different materials inside the patient's body. And whatever the patient responds to, you know he's allergic to, and whatever he doesn't respond to, he's not allergic to. Could we mirror this concept also into treating cancer? Could we actually use nanotechnology to dose patients with minuscule amounts of different medicines and then get a result that would be patient-specific? And what we did is we took those same nanotechnologies I showed you that already I read about as an undergraduate student, and we loaded them with a very small amount of medicine and a barcode, a DNA barcode that enabled actually understanding which medicine we had in each one of these nanoparticles. So the amount of drug in each particle here is enough to kill only a single cell. So it's a very, very low dose. And what we can do is we can take different nanoparticles, each one loaded with a different type of medicine and a corresponding barcode. We can inject them intravenously to the patient. They'll reach the tumor site. And now we can actually follow their activity inside the patient's body. And so that's what we did. We injected them intravenously. They reached the tumor site. We waited for 48 hours. Each one of the nanoparticles distributed into a different cell inside the tumor. We took a biopsy, and then we looked at the biopsy itself. We looked at the live cells that came out of the biopsy and the dead cells that came out of the biopsy. The live cells contained barcodes of ineffective medicines, and the dead cells actually contained barcodes of effective medicines. So now we knew patient-specific, inside this patient's body, which medicines were actually effective. So first we had to test the technology, and you can actually see here a cut of a tumor. These in blue are the cells, and this is actually the tumor region. And the red area are actually nanoparticles that were injected intravenously and target the tumor, this tumor region. You can actually see the difference between the healthy tissue and the tissue that's of the tumor itself. But if you want to look closer into this region, this is a triple-negative breast cancer cell. It's a subset of cancer that's very hard to treat. And you can notice actually the nanoparticles actually inside one of these cells after being injected intravenously. So we went on and we tested the concept: could we actually differentiate between effective medicines and ineffective medicines? And when we pulled out the cells from a tumor, we found different profiles. If we looked at the dead cells, for example, you can see the blue barcodes here of a drug called gemcitabine. And they were found primarily in the dead cells and very rarely in the live cells. Or cisplatin here was found about 1-to-1 in the dead cells and in the live cells, and thereon for each one of the medicines we screened. So we were actually capable of understanding the potency of each medicine inside the patient. So for example, if we looked at the dead cells, we found for each 5 dead cells with a barcode of cisplatin, which is another anti-cancer agent, we found 1 live cell. So this is an effective medicine in this case. However, gemcitabine, we found 6,500 dead cells with the gemcitabine barcode inside of it compared to 1 live cell with a gemcitabine barcode. And this is for doxorubicin, which is a third agent. So now we're actually capable of looking at the patient's tumor and understanding what works and what doesn't work inside the patient's body. We distill it down for the physicians in a manner that will actually, it's a potency chart that gives them the potency of each type of medicine inside the patient's body before entering a clinical trial. So we tested that. Would our projection actually prove itself? And we found that the tumors that were treated according to our projection were actually most responsive to the medicine versus those that were treated against our projection, which actually continued growing and proliferated. And I think you don't need to actually be a pathologist to look at this tumor and see that it's a vibrant one, full of cells that are actually active compared to this tumor that was treated according to the prediction, where you see most of the areas or the regions are actually pretty white, meaning that this tumor is in regression. So when you treat according to a prediction, you actually get the best outcome compared when you treat against the prediction. So nanotechnology actually grants you an ability not only to treat, but also to try and derive information from a tumor and understand what works and what doesn't work. And so it was mentioned earlier that we should show a slide that depicts the technology. This is just a short movie of the barcoded nanoparticles. And they're injected intravenously. They enter the tumor through the leaky blood vessels. And then each one of these barcoded nanoparticles actually enters different cancer cells inside the body. It inserts its medicine. It actually is inserted with it. And the barcode. And here we'd probably choose doxorubicin. So we want to take this concept one step further and ask ourselves, here until now we were taking a medicine and actually delivering it to a patient's body. Could we actually use nanotechnology as a new form for producing patient-specific medicines inside a tumor? Could we look at nanotechnology as millions of factories that are injected intravenously? They circulate in the body, and only where a medicine is actually necessary, we could turn on that factory and produce a medicine of interest for the patient inside the body. And we created particles that had all the molecular machinery that's necessary for decoding DNA and producing a protein. So it actually goes all the way from DNA to RNA and then into protein. And also contains a switch to turn on these particles inside the body so they produce a protein of interest inside the disease site and not elsewhere in the body. And our concept was something like this. The particles would be injected intravenously. They're inert to the body until they're activated. And then where they're not necessary, the particles are just degraded by the body. But where the medicine is necessary, we activate the particles, and then a medicine will actually be formed at the disease site. So the first particle we were capable of producing, this is about a 20 micrometer particle. And you can actually notice the walls of this particle and a protein called green fluorescent protein that is produced inside this particle itself. So the particle contains everything that's necessary for decoding the DNA code into an RNA and then in forming synthesizing the protein inside the particle. A student of mine was actually talented enough to take a movie of this particle producing the protein, having the protein produced inside the particle over time. You can actually notice the green color forming inside. And here it's about 45 minutes. Today we know this process occurs over about 2.5 hours in which the particles continue producing more and more protein inside of them until they're just out of juice and they just can't produce anymore. So we were capable of going down into the nanoscale. Those are microparticles, and we were able to go down to about 170 nanometers in diameter. But when we went any smaller than that, we actually got particles that weren't capable of producing any protein. And we got a ton of criticism about this, saying that it's impossible. Bacteria usually is about 1 micrometer in diameter. How can we go so small and still produce proteins? And then we were lucky enough to come across a paper that came out of NASA. They were looking at the origin of life, and they found nanobacteria that were about 170 nanometers in diameter, and they actually show mathematically that that's the lower limit of life, meaning that a system that would be any smaller wouldn't be capable of actually producing protein off a DNA code. And the reason is that it's just very hard to pack the DNA into these small particles. It becomes mathematically almost unfavorable for the DNA to actually pack into such small confined spaces, but we heard more about that this morning, and I wouldn't try to challenge the math now with the experts we had earlier on today. But this is the particle that we formed, and you can actually see it has a very similar structure to this nanobacteria that was discovered by NASA. But these particles still lack the ability to turn on, and we added just a chemical trick here. It's a switch that sits on top of the DNA molecules. It's responsive to light, and when light at a specific wavelength hits it, this switch is let off, and then the DNA can be decoded to RNA and decoded into protein. And you can actually see it here. So it's not a perfect system. It has some leakage, but it still produces a nice amount of protein when it's exposed to light. And when we tested it also in animals, we were able to actually expose this region to light and then produce a lot of protein inside the specific area we wanted and not elsewhere in the body. So when we talk about nanotechnology, one thing it grants you is a lot of modularity. Particles can be of different sizes. They can use a different system for targeting, and you can add different building blocks into these systems. So there are a lot of tools you can actually use here. But sometimes, as it happened to all of you too, science takes a turn that's unexpected. And so this is the vision we had initially, that nanoparticles would be injected intravenously. They'd reach different sites, and we have these fancy factories inside. And this has been great also for research, and we're continuing that avenue. However, about 1.5 years ago, I was giving a talk at a pediatric cancer conference, and at the time, a person, today a colleague of mine, Dr. Josh Schiffman, was giving a talk about their discovery that elephants don't get cancer. And elephants are remarkable animals, and besides that their body weight is, their body mass is about 100 times our body mass, and that they live as long as we do, they rarely get any cancer. And this is called the Peto's paradox, actually. When you look at elephants throughout evolution, they must have had something we don't have inside their body to protect them against getting cancer. And when you look into their DNA or into their genetic code, you actually see one very, very clear difference between us and between the elephants, and it's a gene called P53, the guardian of the genome. We have 2 copies of P53, and elephants actually have 40 copies of P53. And so this is Josh and I. One of the nice things is that we can actually use, well, I shouldn't say this on tape, but yeah, we can go to research facilities and look at the interesting animals, right? So, well, again, going back to the biology here, if humans have one copy of TP53, or 2 copies of P53, one from mom and one from dad, elephants will have 40 copies of P53. So the first 2 in the human and the first 2 in the elephant are actually identical one to another, meaning that we and the elephants are very, very close to one another. However, then there's another set of P53 genes. Some of them are slightly shorter, some have different alterations, but they're far more robust than we are. And that's what actually grants the elephant cells their ability to protect themselves from cancer. So when an elephant cell starts becoming cancerous, mutations begin occurring, it actually sends the cell immediately to apoptosis, unlike our cells that may go either to cell repair or to apoptosis. In addition, having all these multiple factors of P53 will protect the elephant cell far more than our cells are. And going back to where nanotechnology can interface, can we take the elephant P53, load it into a nanoparticle, inject it intravenously, and now actually protect our own cells in a better manner against cancer? Or maybe as a vision in the future, could we actually create an elephant P53 milkshake and drink it once a week and be protected as the elephants are? So I just want to show you a couple of results. These are pretty remarkable. We're looking at cancer cells. In this case, it's osteosarcoma, one of the deadliest cancers in children. 40 years with no new medicine, and we were looking at cells that were not introduced with the elephant P53, we call it EP53, versus the cells that were introduced with the elephant P53. And actually notice these cells are vibrant, growing, while these are just dying away over time. And it's actually pretty remarkable also in the data. We went on, we tested this in many different cell types. You can see here, this is a close-up. It wasn't photoshopped, so this looks a little bit like Star Wars. And you can actually see the cell exploding after being introduced the elephant P53 into it. And thereon, we tested it also with pancreatic cancer. And I'll end here. Just again, we call this popcorn because you can actually see on the right-hand side what happens to cancer cells, pancreatic cancer cells, after being introduced the elephant P53. So it's human pancreatic cancer after being treated with a human P53. So just a take-home message regarding nanotechnology materials in therapy is that in every case, we actually want to go and adjust those systems for the specific care and for a specific treatment. We can do it either in one way. It can be for retrieving data from a patient. It can be for fixing something inside a patient. And maybe later on also maybe introducing something we're missing inside a patient's body and taking something from nature, such as an elephant gene, and actually adding it onto us so we can protect ourselves in a better manner. Thanks a lot.